Thank you, and thank you, Kate, for that very nice introduction. Today, I'm very excited to share with you or talk to you about a project that I've been working on that's been looking at a possible mechanism that's gating the transition to behavioral habits, which are a very important part of our everyday behaviors. And there are generally two processes that guide our behavior. One is goal-directed actions that rely on the learned association between an action and its outcome, meaning that the performance of that action requires the evaluation of the consequence of that action. The other process are habitual responses that can become automatically triggered once an action has been performed repeatedly. And so these habitual responses are no longer associated with a consequence, so they're just performed without thinking about that consequence. So for example, early in college, I learned that if I wanted to stay alert, I would drink plenty of coffee to study for all those finals I had. And so I repeated this very often, and it worked very well. However, I also learned that if I drink too much coffee, I could get shaky, probably get insomnia. So if I'm goal-directed, I'll think about those goals and consequences and either drink more coffee or limit how much coffee I will drink for the day. However, again, I've now through college, grad school, and postdoc, I've done this repeatedly, so it's really become habitual. And so now I'll drink coffee without thinking about the consequences, and really just the smell of coffee will trigger me to drink coffee, and sometimes too much coffee. And so this is one habit that I don't think, at least not yet, is pathological for me, but some habits can become pathological. And so the balance between goal-directed and habitual actions is really important to guide healthy behaviors, but an over-reliance on habits can actually trigger pathological states such as drug addiction, which you can see why it would be important to understand the neural mechanisms that underlie the transition to habitual behavior. And so goal-directed actions and habitual responses are generally known to be, to require dorsal striatal function. And so the dorsal medial striatum in particular is required for goal-directed actions, while the dorsal lateral striatum is required for habitual responses. And so generally, a lot of this work has been done with lesion studies or inactivation studies, but there's very, very little known about what the molecular mechanisms are that allow the development of these types of associative memories. And so it's widely accepted that transcription is necessary for any learning event to be stored into long-term memory. But transcription doesn't just occur on naked DNA, it actually occurs in the context of chromatin, which is regulated by chromatin modifying enzymes, which then regulates the transcription that's necessary for stable cell changes, or what the yeast literature is the yeast literature describes as cellular memory. And these very stable changes are thought to underlie the stable changes that are required for long-term memories, or at least that's one of the reasons why we started looking at the role of these mechanisms in long-term memory. And so chromatin organizes DNA, and it plays a very crucial role in transcriptional accessibility. And so this chromatin is made up of the basic repeating unit, which is the nucleosome, which actually is made up of histone proteins that have these amino terminal tails, and this interacts very tightly with the DNA. And so again, this organization allows DNA to be compacted. And so in order to get access to this DNA, there are mechanisms designed specifically for that, which one is to modify the interaction between the DNA and these amino terminal tails to relax or repress the chromatin state. And so one of these modifications, and one of the best studied, is histone acetylation. So here's a cartoon diagram of the repressed chromatin. So it's the DNA here in this bluish purple interacting with the amino terminal tails. And so if you add an acetyl group, which is carried out by histone acetyltransferases, you can relax the chromatin state and produce this more transcriptionally permissive state. Histone deacetylases, on the other hand, remove these acetyl groups and return chromatin to this repressed state, which is associated with transcriptional repression. And it's known that drugs of abuse, stress, learning itself can trigger this acetylated state. And several studies have looked at the role of these mechanisms in learning and memory, including myself, as Kate mentioned. And again, the reason is that these enzymes have been known to regulate very stable cell changes. So what we did, or what I did previously, was look at the role of a histone acetyltransferase in the development of context drug-associated memories, which is thought to be a form of a very stable memory. And so what I did was use CBP flox mice in combination with Cre recombinase to delete CBP specifically in the nucleus accumbens. And so if we delete CBP, or the histone acetyltransferase, we'll push the system to this more repressed state. And so when you train these animals to learn a context drug-associated memory by pairing cocaine with a specific context, what you see is that the animals that have a deletion of this histone acetyltransferase are unable to form that drug-associated memory. And so from this, we see that you need the transcriptional activation in order to be able to form these types of memories. Conversely, once you form those very robust drug-associated memories, there's another process, extinction, which is another type of learning process. And extinction itself is not very robust. It's dependent on learning, but it's not very robust, and it's prone to reinstatement. And so what we thought was, well, if you need acetylation to form a memory, maybe if you push the system to this more relaxed chromatin state, producing transcriptional activation, maybe you can enhance this type of learning. So that's actually exactly what we saw. So once the animals form this drug-associated memory, shown as this preference for the drug-paired compartment, we then extinguish this and administer the HDAC inhibitor immediately after each training session. And you see that the animals that were treated with the high dose lose their preference for that drug-paired compartment almost immediately. And when we then challenge these animals in a reinstatement test, you see that these animals that were treated with the HDAC inhibitor during extinction learning do not reinstate their preference for that drug-associated compartment. And so this shows that if you block these histone deacetylases, either non-specifically or with a specific HDAC inhibitor, and push the system to this open chromatin state, you can enhance extinction, not only the rate of extinction, but also its persistence. And so, again, this shows that these enzymes really do regulate these stable changes that are very long-lasting. And so this makes it a very appealing target for habits that are very stable. And so again, most of the work has identified some of the neural substrates that are necessary for the development of these actions or habits. However, we don't know what the molecular mechanisms are. So we wanted to target, or we asked the question, whether histone deacetylases regulate the learning that underlies either actions and or habits. So in order to study whether animals are goal-directed or habitual, we trained animals to press a lever for a reward. And immediately after each training session, we administered the vehicle or HDAC inhibitor, sodium butyrate. And so we can't really tell whether animals are goal-directed or habitual just by lever pressing. So what we have to do is examine whether they're evaluating the outcome of that action. So what we do at test is use sensory-specific satiety, which means that we're giving the animal unlimited access to that outcome, which will generally produce disgust. And so then we use a probe test to evaluate their lever pressing performance. And so if an animal is goal-directed, they will think about that outcome and maybe generally remind them that it'll be disgusting, and so they will not press on that lever if the outcome has been devalued. Alternatively, if the animal is now habitual, they will no longer be thinking about that outcome and continue to press that lever even though it's been devalued. Okay, so with limited training, what you'll see here, and all of the data will be presented in this manner, so I'll walk through the first one rather slowly, but what we have is just press rate on this axis over training days, and you see that the animals acquire the lever press task, but then at test, what we do is normalize their lever press activity because these are within subject measurements, so it will normalize the valued, or their lever pressing during the valued state divided by the total lever pressing on both tests, and vice versa with a devalued state. So this, for the vehicle-treated animals trained with limited amounts of training, you see that these animals are sensitive to the outcome devaluation, so they decrease their lever pressing in response to the devaluation. With repeated training or with extended training, animals will show insensitivity to outcome devaluation, so they'll press the same whether the outcome is valued or devalued, indicating that these animals are now habitual. And so what we see with the HDAC inhibitor treatment immediately after each training session is that with limited training, there's no difference, or with extended training. So this indicates that yes, they can acquire the task, and they could, or at least they have that action-outcome association intact. With limited training, with extended training, we really see no difference. But what's the really interesting part is with this intermediate training, at a point where the vehicle-treated animals are still goal-directed, those animals treated with the HDAC inhibitor are now showing insensitivity to outcome devaluation. And so this shows that the HDAC inhibition is accelerating the rate at which animals transition to habitual control of behavior. Oh, this is cut off at the top, but we went on and tested whether animals, whether HDAC inhibition will accelerate habits by using a slightly different assay. So instead of devaluing the outcome, we sever the association between the action and the outcome. And we see a very similar effect in which the animals treated with sodium butyrate do not show a difference whether the animals are a yoked control or whether the contingency between the action and the outcome has been reversed. And so so far, all the animals or all the data that I've shown you has been from animals that have been trained with a random interval schedule. And so random interval schedules generally will produce habits much more quickly. And so what we thought was, well, if you're using an HDAC inhibitor, what it normally does is that it creates this permissive state. So it'll just allow whatever you're teaching the animals to do. And so what we did was then switch to a random ratio schedule, which actually maintains animals in a goal-directed behavior for longer. And so when we do this, when we train animals on this random ratio schedule, you still see that the animals treated with the HDAC inhibitor are showing insensitivity to outcome devaluation. So they're still habitual, even with this intermediate training, and even though we're using a training schedule that should be keeping them goal-directed for a longer amount of time. Importantly, what we also showed was that if you administer the HDAC inhibitor well outside of the consolidation window, these animals will remain goal-directed. And so this shows that HDAC inhibition, or this shows that histone deacetylases are negatively regulating the consolidation of habits, as opposed to some other non-specific effect. And so far, what I've shown you is that if you use sodium butyrate, you can push the system to this more relaxed chromatin state to accelerate the formation of habits. But what we wanted to know was where, because all of these experiments have been with systemic treatments, so we wanted to then go in and see, well, where in the brain are these changes occurring? And so using fluorescent immunohistochemistry, what we did was look at histone acetylation after that last training session, the last intermediate training session where we saw our behavioral effects. And so I looked at several different brain regions that have generally been involved in the acquisition of these types of behaviors, but the only place that I found a significant difference was in the dorsal striatum, both in the dorsolateral striatum that's known to be required for habit, and the dorsal medial striatum, which is generally or canonically known to be associated with goal-directed behavior. And so given that we saw this increase in histone acetylation, which should be associated with a transcriptionally permissive state, we then looked at gene expression within these brain regions. So in collaboration with Pam Kennedy, what we did was look at several different genes. We started off with a panel of immediate early genes, and it looks a little bit busy, but generally, so we looked in both the dorsolateral striatum and the dorsal medial striatum in home cage control rats and intermediately trained rats with either vehicle in the lighter colors or sodium butyrate in the darker colors. And generally what this is showing is that there's an increase in the expression of these immediate early genes in both the dorsolateral striatum and the dorsal medial striatum with no difference induced by the HDAC inhibitor treatment. So generally what we can say from this is that activity in these brain regions is being induced, but what we did see differences in were a few other genes. One was ATF3, which is part of the Krebs superfamily that's known to be involved in learning and memory. And so with the HDAC inhibitor treatment, you see this very robust increase in its expression in both the dorsolateral and dorsal medial striatum. Another interesting target was DARP32, which is related to striatal plasticity. And what we see here is that in the dorsolateral striatum, you see an increase with the HDAC inhibitor treatment, a little bit further increase than just training alone. And in the dorsal medial striatum, in which training alone reduces the expression of DARP32, with the HDAC inhibitor treatment, you prevent that reduction. And so this was interesting, and especially the DARP32 finding, because it is associated with both dopaminergic and glutamatergic plasticity in the striatum. And so the thing about DARP32, though, is that depending on how it's phosphorylated, its activity within a cell will change. And so, again, with collaboration with Pam Kennedy, we looked at the phosphorylation state of DARP32 and found that at the threonine 75 phosphorylation site, there's an increase in those animals treated with the HDAC inhibitor, but we didn't really see anything at the phosphorylation of threonine 34. And so this starts to indicate that maybe the cellular function within the striatum is starting to change. We're actually following up on these findings in collaboration with Dr. Mike Levine and Lori Galvan, that's where we'll be looking at whether there's any changes in the physiology within the striatum. So what we can conclude from that piece is that HDAC inhibition accelerates habits, and there are changes throughout the dorsal striatum in both acetylation and transcription. And we know that the dorsolateral striatum is required for habitual responses. And so what we wanted to do was then target these brain regions to see whether if manipulating histone acetylation specifically within these regions, you can then alter behavior. And so we started off with a dorsolateral striatum. But one other thing that we wanted to then hone in on was that sodium butyrate is a non-specific HDAC inhibitor. It generally targets class 1 HDACs within, it generally targets class 1 HDACs, and within this class of HDACs, HDAC3 is the most highly expressed HDAC in the brain. And so what we did was use specific HDAC3 manipulations within the DLS to then examine what behavior, how behavior would change. And so what we did was train animals, again, using this intermediate training, and infuse RGFP 966, which is a selective HDAC3 inhibitor, into the dorsolateral striatum immediately after each training session. And so this increases histone acetylation within the dorsolateral striatum. And since this is increasing histone acetylation, creating this permissive state, the prediction is that you would enhance habits. And so that's exactly what we see with this intermediate training. When vehicle-treated animals are normally goal-directed, the animals treated with the HDAC3 inhibitor, specifically within the DLS, are now showing habits. We went on to then use a different manipulation using a viral vector to express a point mutant of HDAC3, which renders HDAC3 inactive. And so expressing HDAC3 within the dorsolateral striatum here, if we look at histone acetylation within this region, you see that you get this increase in histone acetylation compared to other regions that do not have the HDAC3 point mutant expression. And so again, this increase in histone acetylation transcriptionally permissive state, again, you see that these animals are showing insensitivity to outcome devaluation. So again, two different ways of approaching the same enzyme, and they both show that you can accelerate habits. And so or at least together, this shows that HDAC3 is normally repressing some type of mechanism to prevent habits from forming. And so if that's true, if you then overexpress HDAC3, you may be able to prevent habits from forming if HDAC3 is normally repressing habitual behavior. And so for this, what we did was overtrain animals. So we did extended training and now use viral vector to overexpress HDAC3 in the dorsolateral striatum. And so this produces a decrease in histone acetylation. And so this transcriptionally repressive state, again, the prediction is that you would prevent habits from forming, and that's exactly what we see. So now the control animals with the empty vector are showing insensitivity to outcome devaluation because of this extended training. And the animals that have the HDAC3 overexpressed in the DLS are remaining goal-directed. And this is true even if we extend training even further. These animals still remain sensitive to outcome devaluation, indicating that they're still goal-directed. And so from this, what we think is normally happen, again, is that HDAC3 is normally repressing transcription that's necessary for learning. And so when you remove HDAC3, you can get the transcription that's necessary for the formation of habits. And so this was in the dorsolateral striatum. But remember, we also saw changes in the dorsal medial striatum. But the dorsal medial striatum is canonically ascribed to goal-directed actions. And so what we wanted to do was see if we manipulate these exact same mechanisms in the dorsal medial striatum, what would happen to behavior. And so this was actually surprising. So again, exact same thing, but in the dorsal medial striatum, we inhibited HDAC3, got this increase in histone acetylation. And so given that the dorsal medial striatum is required for goal-directed behavior, one might predict that goal-directed behavior would be enhanced. But what we actually saw was that those animals are also showing insensitivity to outcome devaluation. So they're also becoming habitual at a faster rate, even though this is the dorsal medial striatum. And we repeated this again with the HDAC3 point mutant, and we saw the exact same thing. So these animals, again, are showing habitual behavior at an earlier time point when we're producing this transcriptionally permissive state in the dorsal medial striatum. And so, again, the same question is, what would happen if we then overexpress HDAC3 in this brain region? What we found is that this decrease in histone acetylation within the dorsal medial striatum also prevents the animals from acquiring habitual behavior. And exactly the same if we do extended training, these animals still show a lack of habitual behavior. So they remain goal-directed. And so it appears that HDAC3 is repressing the transition to habits in both the dorsal medial striatum and the dorsolateral striatum. And so just to conclude, HDAC3 is a negative regulator of habit formation, and again, both in the dorsal medial and dorsolateral striatum. And it appears that HDAC inhibition is producing a gene expression profile that may underlie changes in synaptic function within these brain regions to ultimately change behavior. And importantly, what we think is that dorsal striatal HDAC3 is normally preventing habits from forming. So it's acting as a molecular brake that remains in place and prevents habits from forming too early. And under the suitable conditions, or such as repeated training or other things, habits can then form and dominate behavioral strategy. And what's interesting about that is that things like stress and drugs can actually trigger these same mechanisms. They can cause a hyperacetylated state, and those both stress and drugs have been shown to potentiate habits. So it'll be interesting to see if this is really a mechanism that is playing a role in that type of accelerated habit. And so with that, I'd like to thank, of course, Kate, that provided this great opportunity to do this project, and Vinu Screenfield, that's helped me with a lot of the behavioral, or carrying out a lot of the behavioral experiments. And of course, Michael and Christine, that have helped me just generally with everything. Everyone in the Wassum lab, Pam, that's helped with all of the gene expression studies, Marcelo at UC Irvine, that generously provided the viruses to manipulate HDAC3, and of course, Mike Levine and Carlos and Lori, that will be helping us with the ePhys experiments and all the funding, and of course the award, the Scheibel Lecture Award.